Organ shortage is a major problem in transplantation. There are more than 7,000 patients on the waiting list in the UK. This has led us to consider ways of increasing the number of available organs, perhaps by considering donors that are older or who have other medical problems. But as clinicians, that decision about whether to use an organ or not can be a really difficult one. And we know it's important to get right because our decisions are life-changing for the patient. So one way we can assess organs is to put them on a testing rig, and this gives us the opportunity to take samples from them. We've been working with Andy Fisher and colleagues in Newcastle to take lung samples and to test them to try and identify markers that allow us to predict whether an organ is going to work well when it gets into the recipient. So the tests that we've been using are based on this molecule. So this is DNA, the genetic code that's found in every cell. So let's go to the lab and find out how we've been using this test.
Genetic material comes in different flavours and performs different roles.
DNA is the instruction manual for life, giving organs all the information they need to function. These instructions are passed on in the form of instruction sheets called RNA, and it is these bits of RNA which are used to make the machines or proteins that enable a cell or organ to function. At present, the technology for assessing RNA is more advanced than for proteins, so this is what we chose to measure. We examine the relative levels of different RNAs in tissue taken prior to assessment for suitability for transplant on the testing rig, and compare this to the outcome data. First, the RNA is extracted, the sample prepared, and the RNA sequenced. We then look for different levels of different RNAs between organs which are deemed to be suitable for transplant and those that are not.
We hope that through this work we'll be able to better identify organs which are suitable and so increase the number of patients that are able to be transplanted.